Welcome, my name is Nick Berry from the University of Southampton, and this small instructional video is to help you use our computational tool called BARMIE. This work was funded by the Natural Environment Research Council from a grant to myself and Matt Lucucci at Brunel University. BARMIE stands for Binding Affinity Ranking at the Molecular Initiating Event. The molecular initiating event comes from the adverse outcome pathway concept, where a chemical's interaction with a protein causes a cellular response leading to an adverse outcome. Our research is trying to see if we can use data from the sequence genome information to identify which species may be most sensitive and those chemicals are most concerned based on the interaction with the MIE. The work is in conjunction with Matt Lube, Ali Pandini and Parsa Fuladi at Brunel and Fernando Calajaro Nunes. The GitHub script and instruction documents can be downloaded from the manuscript here at bioRxives, and we've used OpenBabel, PyMol and AutoDocVina in generating this script. The first phase is to obtain your proteins. We've used UniProt. UniProt is linked to the genomes in Ensembler and AlphaFold to predict the structure of proteins. For this exercise, we'll use the fish glucocorticoid receptors NR3C1. We initially get a lot of hits, and thus we use advanced tools to obtain those for teleosteae. The 660 results will include all isoforms and truncated predicted protein structures. At this stage, we do not filter. To download, we go to the download option, compressed no, and we want an Excel file. Once downloaded, it is important to change the name to UniProt. For the chemical retrieval, we've used PubChem in this instance, but any other chemical structure download program can be used as long as the file is in an SDF format. For this, I will just use dexamethasone as an example. PubChem will provide details of the chemical structure and the drug, and we need to download the 3D conformer as an SDF file. So moving on to setting up BARMIE. I'll split my screen into two with the HPC terminal on the left, which contains the BARMIE main folder. This can be downloaded from the GitHub site, and you might need your institutional IT wizard to help you upload the file. On the right, we have the instruction manual that can also be downloaded from the GitHub site. Once installed, open the file BARMIE, go to the scripts folder and create these 6 folders illustrated. Be careful to make these the precise names, as this is what the script will recognize. We then need to inform the HPC which directory we are using, and we simply do this via cut and paste. We also need to ensure that we're in the PyMol environment. This instruction is number 3 on the instruction manual, and you just cut and paste the instruction. Once this is done, we then drag in our UniProt file, which I have already done on the left hand side, and then we go through running the information on the instruction manual. I've already done this, and within the PDB files you'll see a list of all of those 660 or so files. We now need to go into the next phase, which for us is to filter the GRs. This script line is specific to GRs. For those unfamiliar with steroid receptors, they are modular proteins. The AB domain is disordered. We have a DNA binding domain and a hinge region, and the final domain being the ligand binding domain. For this exercise, we have chosen to look at just the ligand binding domain. This region is highly conserved, and we know the conserved amino acids, so we have instructed the program to find the amino acid sequence KAIEP and then cut the sequence 7 amino acids further along. This obtains us the ligand binding domain. You'll need to change this instruction to suit your receptor or protein. You can leave this step out, but you need to ensure the script reads from the correct folder in subsequent steps. The simplest way may be to just copy the files from the PDB file folder and paste into the PDB files filtered folder. The next step is really important. If manually using AutoDocVina, each structure is uploaded in a particular orientation. You then set coordinates and size of the docking box. I'll explain this later. This orientation will differ for each protein. To overcome this, BARMIE uses the alignment tool in PyMol to ensure all the protein structures are orientated the same. For this, it doesn't matter which sequence you align to, they are all conserved. We have used AF-P4984-F1-model_v4.pdb. Once this has occurred, we then need to ensure that we convert the aligned protein and also the chemical structure to a PDB-QT file, and these are recognized by AutoDocVina. Please ignore the warning signs. This is a glitch in OpenBabel and does not affect the docking process. At the end, you can see that we now have 180 PDB files converted to PDB-QT. And now we just need to do the same for the ligand. Obviously, you can have more ligands within your ligand SDF folder. As I said, I am just using dexamethasone as an example. Once this is complete, we are then ready to perform the docking program. But first, I will show you how to set the coordinates. This is the only manual stage of the whole operation, and you need to know a little bit about your structure and your the binding pocket. For this, I'm using the salmon GR, and I bring in, just drag in the PDB-QT file. If we go down, we can see that this is actually the full length structure with the AB domain as well, and we have this box that sits over the protein, and we want this box over the ligand binding domain. To do this, we have set the box size at 20-20-20. And for the GRs, we know of these 3 helices, and in this instance, the binding pocket is towards the bottom on the left. And and we simply move the box around to that region. Now I'm not going to do this, but what I would recommend is that you bring in the ligand, that you bring in that dexamethasone molecule, and hit run, and you'll get a binding affinities derived. We then go back and tweak the X and Y, Z coordinates until we find the lowest binding affinity, and then we use those coordinates in the docking. We're now ready to start the docking process. Initially, I would set a output file, so output testing BARMIE. This is where everything will go. We also need to make sure that we're in the correct directory docking. Once we're there, we can open the Slurm file. This has the main script, the docking script. You need to check with the IT department if the code in green is applicable to your HPC. We have the output file as well as the coordinates from AutoDocFina in the main run the Python script. So we have the E, which is exhaustionless of 32, and then the coordinate center X equals 5, Y equals 2, and Z equals minus 15, and then B, the box size. Once we've done that, we need to change from the PyMol environment into the VINA2 environment. And then we need to ensure that we're in the docking directory by cutting and pasting the directory name on the left hand side. The instructions give a bit more detail on the Slurm file coordinates, etc., and also on the choosing of the coordinates. We're now ready to run the Slurm file. The next stage is to download the data. It's important to make sure that the file names are correct. As you can see, I've made quite a few mistakes above, but we just follow the instructions. So cd space double dot cd scripts, and then we'll take the instruction from the instruction pages, cut and paste that, and then directly copy and paste the directories that we require. So the first one being the output file, where it's all gone. And the second one is where you want your Excel file with the results in to be located. I've simply just done this in the BARMIE main file. And then call it whatever you want. I've just done test.csv. Locate the file and then open. And here you can see that we have the file name, which is the UniProt ID identifier, and the binding affinity minus the -8 to 9 numbers.